Okay, good morning once again, my dear students. As we already started the mitosis division, and now we are going to explain how the division takes place. So, the mitosis division, it's occurring to two phases. The first, that is interphase, and the second step, that is M phase or mitotic phase. Now if we talk about the interphase, so interphase is a phase in which the cell prepare itself for the division, and by the division it's produce the daughter cells. So that is, we consider it is just a preparatory phase, and during this phase there is no any visible changes observed in the cell. Only what happens in this phase? The cell is much metabolically active, and because there is no visible changes is observed, so this phase is also considered as what? It's also considered as resting phase. Means interphase is also considered as resting phase. Why? Because there is no visible changes observed in the cell. Only the cell is much metabolically active. It's prepare itself, and this interphase can be divided into three subphases. The first one is G1 phase. It's also known as first growth phase. What is first growth phase? The first growth phase is a phase in which the RNA proteins are synthesized in the cell. Their volume is increased, the cytoplasm is increased, and if it is animal cell or plant cell, then the mitochondria divide and it's produced in the cell. If it is plant cell, then chloroplast also divide and produce the duplicates copies. Means during the G1 phase, the most important thing what happened? The RNA synthesize, protein synthesize, and all the cell organelles, it's multiply in their number. Volume of the cell cytoplasm, it's also increases. Clear? Now the second step of the interphase, that is S phase, or it's also called synthesis phase. This synthesis phase is very important. Why it is very important? Because during this phase, the genetic material of the cell, that is what? That is DNA. So DNA is replicated here and it's multiply, and this process is known as replication of DNA, or we can in generally say the synthesis of the DNA. So the synthesis of DNA, it means the synthesis of the genetic material takes place during the synthesis phase. That's why it is considered as what? S phase. Clear? Now the next phase, after the completion of the synthesis phase, the next one, that is G2 phase. It is also considered as second growth phase. It is very short growth phase. Means the synthesis of DNA stop in this phase, but the synthesis of the RNA proteins, it's continue, and all the contents inside the cell is increased in their number, and now cell is totally ready for the division. Clear? So interphase, which is also considered as resting phase, divided into G1 phase, S phase, and G2 phase. Summarize: G1 phase, it's a synthesis of the RNA and the protein, multiplication of the cell organelles. Synthesis phase, S phase, there is replication, duplication or synthesis of DNA, and G2 phase means the cell is totally ready for the division. Clear? Any doubt? No. Now, next, after the completion of the interphase, the cell enter into the next phase, and that is M phase or mitotic phase or mitosis phase. When we see the mitos means thread. It's referring the chromatin thread or chromosomes, and this mitotic phase is completed into two steps. The first one is karyokinesis. Karyokinesis, it's derived from the Greek word karyon. Karyon means nucleus. So karyon means nucleus and kinesis means movement or division. So this phase, karyokinesis, means it is the division of nucleus, and it's completed into four steps, four phases. We discuss it later. And the second, after the completion of the karyokinesis, it's followed by the cytokinesis. Cytokinesis means the division of the cytoplasm. So after interphase, there is two steps. First one, the karyokinesis, and the second one, cytokinesis. Karyokinesis means division of nucleus, and cytokinesis means division of cytoplasm. Clear? So the process of the division of nucleus, and this karyokinesis completed by the four phases. That is, first one is prophase, second one is metaphase, third one is anaphase. Now, karyokinesis, I already told you, it is what? Division of nucleus. And this karyokinesis completed by the four phases. The first one is prophase, second one is metaphase, third one is anaphase, and fourth one is telophase. Among these, this anaphase is very important, and for better understanding, these phases can be further divided into three sub-phases like early prophase, then middle prophase, and then late prophase. It's not given in the syllabus early, middle or late, but for better understanding, we can say early prophase, middle prophase, late prophase. After that, early metaphase, middle metaphase, late metaphase. Same here also, the early prophase, early anaphase, middle anaphase, late anaphase, and then the early telophase, middle telophase, and late telophase. So this is what? This is the steps by which the nucleus of the cell divided. Which one? Prophase, metaphase, anaphase, telophase. Clear? Is there any doubt? Now come to the prophase. But before the starting of this prophase, it is better to know about the structure of cell, and then we start. If any cell, if we take any cell, suppose we take animal cell. So what thing is present in the animal cell which involve in the karyokinesis? The outermost protective covering, that is cell membrane. Inside the cell membrane, cytoplasm, and in the cytoplasm there is nucleus. Double membranous bounded structure. Double membranous bounded structure means the nucleus is bounded by 2-unit membrane. Clear? So, nucleus is bounded by 2-unit membrane. So, outermost covering, that is called nuclear membrane. Then the protoplasmic part inside the nuclear membrane, that is nucleoplasm. Then the condensed form of the genetic material, that is considered as a genetic material, that is chromatin network. It is what? Chromatin network. And one dense portion, that is called nucleolus. And in one side of the nuclear membrane, outside in the cytoplasm, there is a cell organelles called centrosome. Which is responsible for the division of the cell. So, if we talk about any animal cell, so these structures are involved in the cell division. That is what? The cell, outer covering, that is cell membrane, cytoplasm, then nucleus, this is completed nucleus, and the nucleus contain protective covering, nuclear membrane, inside the nuclear membrane nucleoplasm, then chromatin network and nucleolus. And if we talk about the plant cell, so all the structure is same, another structure is same, but there is no centrosome, and one more protective covering is present around it. You already aware? That is what? That is cell wall. Clear? So this is the cell which is going to divide. It means this cell is going to divide by the mitosis division, enter into the karyokinesis, and prophase begins. Okay?